Our next company is Nininger Medical. Andrew, I believe, will be presenting.
Good afternoon. I'm Andrew Schombs, co-founder and chief operations officer for Nininger Medical. We are developing a revolutionary leaflet material to be used on next generation replacement heart valves. Leaflets are the part of the valve that open and close that make the valve work. And there are globally approximately 250,000 replacements done each year. Most of those are done through a small tube called a catheter, and that's how it's implanted in the body. The valves, as the one you see up here, are normally made with leaflets derived from animal tissue. That presents some challenges. That pericardial tissue is yesterday's technology. To harvest it, to treat it, to install it into the valves takes a lot of time and effort. The material itself isn't very uniform, and its thickness prohibits the catheters from getting smaller and smaller. Calcification is a reaction where the body ends up making the leaflets stiffer, and by making them stiffer, it actually makes it harder for the valve to work, and it shortens the valve's life cycle. We're addressing this by using nitinol 3D thin film technology. And as you can see, our thin film is significantly smaller in thickness. And by having that, what it does is it enables the valve to be able to be pushed down into a smaller catheter size. And because it's made of nitinol with its non-porous, very smooth surface, it's naturally resistant to calcification. And because it's an engineered material, it can be mass produced to exact specifications for whatever valve design is required. Here's a video of our working prototype. In this video, you can see 3 different leaflets. Each leaflet was made at a different wall thickness. The one in the lower left-hand corner is the one that is more appropriately mimicking what's in the natural valve today. And some of our next steps related to our timeline is to continue working with the manufacturing process to optimize that thickness to get the mobility of the leaflet to be a bit more appropriate for the flow rates that are needed for heart valves. We're hoping in the next year to 2 years to develop a strategic partnership to take this technology and to put it into some preclinical or animal studies with the goal of eventually being in humans by 2027. The global heart valve market is about 17 billion. The slice that we're looking at is using our thin film technology for the valves or for the delivery systems that need it most. So the smaller catheter sizes in terms of its diameter for delivery, or even the biggest valves, such as the tricuspid and mitral valves. Myself and my co-founder and CEO, Dan Anderson, we have over 40 years of combined medical device experience, most of that in cardiovascular. We actually met at Edwards Life Sciences, and I was also working in the Tissue Center of Excellence at Edwards, so I know all about harvesting this tissue. We have a great panel of advisors, scientific and business, from the cardiac space, and all of our business advisors have deep knowledge in medical devices. Again, we're Nininger Medical, and we're developing thin leaflets for next generations of heart valves. Thank you.
Great. Thanks. All right. Thank you, Andrew. I know we have an Edwards representative here, so I don't know if you want to go first, but judges.
Sure, happy to. Great presentation, Andrew. As you very well know, this is something that's directly relevant to a key product area for Edwards. I have 2 questions. One is related to kind of the value proposition or the benefit of getting this on a smaller catheter. Do you have any estimates of how many additional patients or what percentage of patients getting this on a smaller catheter would actually unlock in terms of ones that aren't that are kind of restricted from this type of therapy based on their anatomical challenges today?
Yeah, as far as anatomical challenges, I'm not quite sure what new percentage of patients that this would help. When we started, our goal was to actually develop a transcatheter tricuspid valve. And in the tricuspid valve market, we know that the catheter size itself is relatively large. And the problem is being able to get these larger valve sizes to squish down into that catheter. So if I remember correctly, it's something like 1.6 million patients with tricuspid problems, with a very small percentage being treated today, something on the order of 5%, where this technology was focused at trying to be able to give every single one of those patients a smaller catheter with a better heart valve.
Great. And then my second question is related to, what is the expected lifespan of this material? Have you done any in vivo simulations to kind of project that?
Yeah, we haven't had the opportunity to do those types of simulations yet. That's part of our next round of testing. But this technology is already being used in stents today. And those stents, as you know, would also have to go through cardiac rhythmic cycles. Our manufacturing partner that has helped to develop that technology, they have been able to provide us some data on its long-lasting fatigue properties. Unfortunately, I don't know exactly how many millions of cycles they went out, but I know it was quite large. Thanks.
Can you talk a little bit more about the cost savings that are expected?
Yeah, so tissue today, when it's harvested, usually from cows, right? It's sort of a scrap material, but the processing that comes after it is quite large. Oops, sorry, is quite large. And then the actual act of sewing it into the valve, right? So with this technology right now, because we're doing things on a small scale, the leaflet costs at the moment are quite big. But what we're aiming at is trying to reduce the cost of the overall assembly of the valve by making these leaflets pretty much ready to be transferred to the valve. We're working on attachment methods right now to figure out what's the best way to do that. And so from a cost perspective, we're thinking that we could either come in about 20 to 30% lower, or depending on that attachment method, it might even be more than that.
Great.
We spoke at lunch, so but for the sake of the audience, I ask the questions again. Could you talk me through the IP portfolio? I know there's a patent pending. Nitinol is, like you mentioned, this is a very common. It's not a novel material for cardiovascular.
Right.
So just trying to understand what's the claim of the patent, how deep is the IP protections and defensibility there.
Right. So I'm not an expert in the IP field myself. However, our claims are associated to the fact that it is a thin 3D shape. The existing or the prior art that's out there today does deal, obviously there's lots out there for nitinol, especially in med devices. And there is also the use of this thin film, but all of the previous uses of the thin film is to make it in a two-dimensional shape and then apply it to something else. Whereas this patent is focused on being able to construct the exact geometry that would be needed for the heart valve to distribute pressures appropriately for whichever valve that it's being used for. And the claims are around its application, not that it's nitinol is new. It's the application of it that's new.
Got it. Follow-up questions there. Is there different in the way a surgeon would implant this? Is there a different procedure?
No, it wouldn't change the procedure at all. It would use the same type of transcatheter-based technologies. The only difference is there would be theoretically, sorry, I don't know the numbers, more access to patients that have more difficult vasculature, or if you're replacing like a large valve, like the working prototype that we did was based off of a 50 millimeter valve, and those 50 millimeter valves just can't get inside of a small enough catheter to compare to what is a typical transaortic valve today, which is closer to 16 French. We would be able to get this 50 millimeter valve down to about 22 French, if I remember correctly.
Got it. Thank you.
Andrew, great pitch. What kind of preclinical data do you have right now? Is it animal model testing? Or?
We don't have any animal model testing as of right now. Yeah, the video that you saw was us fixturing the leaflets into a hydrodynamic pressure tester that was using the appropriate pressures for in the body, actually slightly elevated, but we don't have any clinical data yet, or preclinical data, sorry. Yeah, that's our goal for 2024.
What's the regulatory pathway? What's the regulatory pathway?
Yeah, we're still working through with one of our consultants on how that would work, because right now we're trying to develop the technology of the leaflet. The leaflet itself is not something that would necessarily be a standalone device. So it needs to be integrated into a device, which would then go through that regulatory pathway. But that regulatory pathway would probably look a lot like most other new valves today, where there would be some sort of a PMA or clinical trial.
Okay.
Thank you.
Yeah, thank you so much.